Let's go on to the next one, the second one. Now, the second one's really cool, all right? And this is the type, we're gonna have this one be the type IIx muscle fiber. Okay, so this one is gonna be specifically the type 2x muscle fiber. Now again, type 2x muscle fibers are special because we can also call them another type of name. We can call these ones fast glycolytic. Okay, so these are the fast glycolytic ones. But let's actually, so we can keep it consistent, let's come back to this one because this one's going to make the most sense after we finish up this next one. So let's actually come back to this one in a second. We're going to do this one because this one's actually going to go along with what we did over here. Okay, so this last one that we're going to talk about is specifically called the type IIa muscle fibers. So this one is actually called the type IIa muscle fibers. And the type IIa muscle fibers are really special. And the reason why is that these are fast oxidative muscle fibers, okay? Now, fast oxidative. Now, some of you guys who are out there and you guys lift or you enjoy learning about muscle physiology, you've probably heard the term fast twitch muscle fibers. And you've probably heard of the term slow twitch muscle fibers. So your slow twitch muscle fibers are your type 1 fibers. Your fast twitch fibers are your type 2x and type 2a fibers. Now, what really differs between the type 2a and the type 2x? That's what we're going to talk about here. I want to do this one first, so it's going to make more sense. Okay, think about this. Fast oxidative. Okay, let's go back to the structure. Now, with type 2a, let's look at this again. Look at this in just an overall view. This is the smallest. This must be kind of like intermediate, and this is the big boy. So this is the largest of all the skeletal muscle fibers. So this is the largest of all the skeletal muscle fibers. So this is the largest diameter fiber. Okay, now some of you might be like, oh, Zach, I don't know, that's weird. Why would that be? I always thought that the fast glycolytic ones were the ones that for powerlifting and all that kind of stuff like that. That's true. But in humans, type 2a fibers are bigger than the type 2x fibers, okay? In other animal studies, they found that the type 2x fibers, which they also refer to as the type B fibers, are bigger than the type 2a, but not in humans. So in humans, the type 2x is intermediate fiber diameter, and the type 2a is actually the largest fiber diameter. Next thing, I want you guys to correlate oxidative. Oxidative means that it depends upon a lot of oxygen. Now, that means that there must be a lot of blood going to that area. So this muscle fiber must also have a decent amount of capillary density. And that is true. So if we show again here, let's say here I'm just going to show this capillary network here. Here's this capillary network. And again, the whole concept here is that this is going to have a lot of blood flowing through this actual muscle tissue. If it has a lot of blood flowing through this muscle tissue, what does that mean then? It's going to be able to drop off a significant amounts of oxygen to the tissue. Not only that, but what color is our blood normally if it's delivering it to this actual muscle tissue, arterial-wise? It's going to be a nice reddish color, right? So this is going to have a high capillary density, but I want you guys to go back to that concept I explained before. It can have a high capillary density, but it's such a large muscle fiber that even though it has a large capillary density, it's not going to produce the full reddish color because it would take so much capillary density, so much blood flow through this muscle for it to produce a perfect reddish hue. It is still red, but it's not as red because of its large diameter. So it would take tons and tons of capillaries to make it red. It's more of a reddish, pinkish color, okay? So again, when we describe this structurally, we say it has a high capillary density, but because the muscle fiber is so big, even though there's a decent amount of capillaries supplying it, it would take so much for it to cause the whole muscle to produce a nice reddish color. So it's more of like a reddish, pink hue. Okay, next thing, we're already going to throw this down. It has lots of mitochondria. And this should make sense. And why should this make sense, guys? Because there's the word oxidative. And again, if it has lots of mitochondria, Let's draw a couple mitochondria here. If it has lots of mitochondria, what does that mean that it depends upon? Well, that means, oh, Zach said if it has a lot of mitochondria, it's probably going to depend upon a lot of, oxygen. That is right. So it is depending upon primarily aerobic cellular respiration because of the word oxidative. So again, when you think about this one, it's going to be using glucose primarily, right? And it's going to be utilizing oxygen. And when it utilizes this oxygen, it's going to convert this glucose right through these processes. What is it going to do? It's going to lead to the formation of ATP. So it primarily performs this action by aerobic cellular respiration. So it is depending upon aerobic conditions. But you know what's special about this muscle? He has the ability to switch in between aerobic and anaerobic. All right. Now, the type 1 muscle fibers do have a tiny bit of this molecule called glycogen. But the type 2A are going to have a lot of glycogen. Okay, they have a decent amount of glycogen. Okay, so we'll say like if comparing type 2A versus type 2X, type 2A actually has kind of like in between, intermediate or moderate amounts of glycogen. What is glycogen? So let's say I have a glucose molecule here, and I take out another glucose, and another glucose, and another glucose, and another glucose, and eventually it starts kind of just looking like this. It's a big, big polymer of glucose. This right here is called glycogen. Glycogen is plentiful in moderate amounts inside of these vesicles though. Our muscles, they store it inside of these granules. So I'm going to draw these dots here, and these dots are representing our glycogen, okay? This structure here is called glycosomes. And we have a moderate amount inside of this skeletal muscle fiber. But whenever our body needs certain types of ATP and we don't have as much oxygen available, guess what this muscle can do? It can break down some of this glycogen into glucose. And where can this go? This can go into being oxidized and eventually used to produce ATP. Okay? All right. Now, there's another mechanism that can occur. So again, there's going to be this glycosome. So they do have a moderate amount of glycogen. So we've already gone over the structural concepts. Metabolically, we already said that they depend upon aerobic conditions, but they can do anaerobic conditions. Okay, but to be even more specific, whenever glucose undergoes a specific pathway, there's a specific pathway where it actually gets split, because glucose is 6 carbons. It gets split into 2 3-carbon molecules called pyruvate. And during that process, what happens to the pyruvate? We produce 2 ATP. What is this process here called? This process is specifically called glycolysis. This is really what's happening during this anaerobic pathway. So glycolysis is one of the main things that is occurring during the anaerobic process. And this can come from glycogen. But in the same way, some of this glycogen can get broken down to glucose and then go to Krebs cycle, electron transport chain. But it also can go through glycolysis to produce pyruvate and a little bit of ATP. You know what else is really cool? Skeletal muscles are really cool because they have a special enzyme inside of them. A special enzyme. Look at this enzyme. This enzyme is really cool because it helps us to produce a little bit of ATP very quickly. It doesn't last very long, but it helps to produce a little bit of ATP if we need it. There's a molecule that's very plentiful within your skeletal muscles. That molecule is called creatine phosphate, but I'm just going to denote it as CP. So there we have one molecule. And again, what does CP stand for? It stands for creatine phosphate. This is very plentiful in your skeletal muscles and even in your cardiac muscle too. What happens is creatine phosphate is going to bind onto this enzyme. What is this big blue enzyme here called? This enzyme right here, this blue enzyme, is called creatine kinase. And what creatine kinase does is, it can bind into this pocket. So one pocket, the creatine phosphate is binding. The other pocket is going to be ADP. ADP. Let's do this in a different color. Let's put this in blue. ADP. You know what this creatine kinase does? He binds both of these guys and smushes that phosphate off on the creatine onto the ADP. That's pretty cool, right? So what is he doing? He takes this phosphate here from the creatine and adds it onto the ADP. And as a result, what would you get from that reaction? I would get ATP, right? So I'm going to get ATP. When I produce ATP, it's not very plentiful amounts of ATP. But what's coming out of this reaction? I'm also getting creatine. And creatine will get acted on by another enzyme to get converted back into creatine phosphate. But this is one way that our muscle cells, this muscle fiber, can also produce ATP. And again, this is through the creatine phosphate pathway. So the creatine phosphate pathway. But again, being very, very specific, this cell mainly depends upon aerobic. It's more specifically depending upon aerobic, but it can undergo anaerobic processes. Okay, next thing. Think about it metabolically. Since it's a large diameter muscle fiber, this is where it's a little tricky. That's a little tricky on this one. Because it's a large diameter muscle fiber, it does have, oh, think about the word first before this, fast. What is its contractile speed? Fast. It contracts very fast. So if that's the case, then it's going to have a fast contractility, right? So it contracts very fast. This guy here, here's our myosin. Here's the myosin. And then again, what do you have over here? You have actin. So here's the actin cups, right? And again, what did we say was binding to this? ATP. What was the name of that enzyme that was cutting the ATP? Myosin ATPase. If it cuts it fast, what would that say then for this whole cross bridge and sliding filament theory? That's going to occur faster. If that's occurring at a faster rate, won't the actual contraction speed increase too? Yes. So this fast contractility is due to fast or high amounts of myosin ATPase activity. Okay? So fast or high amounts of myosin ATPase activity. Now, not only is there fast contractility, not only is there fast myosin ATPase, let's think about the word oxidative. That means it produces a decent amount of ATP. So because it produces a decent amount of ATP, okay, it's pretty good at being fatigue resistant. But if it contracts really, really fast, wouldn't it burn out pretty quick? Yes. So it doesn't last hours, but it can last a decent amount of time within minutes. So specifically, this one is fatigue resistant, moderately. I'd say it's moderately fatigue resistant, right? So it's moderate fatigue resistant. But because it has a moderate fatigue resistance, so and like I said, it can last for maybe a little bit less than 30 minutes. So this muscle fiber can continuously carry out its contractile activity for a little bit less than 30 minutes. But it can generate a decent amount of power. So I would say it generates a moderate amount of power. But not as much power as the type IIx muscle fibers. Okay? So again, recapping this, metabolically, it carries out aerobic cellular respiration. It can do anaerobic via glycolysis and the creatine phosphate pathway. It contracts very fast because it has a high amount of myosin ATPase activity. Remember this enzyme that we showed you here? This enzyme that was connected with the myosin. Here's the myosin right here. It has a high amount of myosin ATPase activity. What does that mean? It's good at breaking down ATP into ADP and inorganic phosphate very fast. And also, we said that because it contracts very fast, but it does produce decent amounts of it does produce decent amounts of ATP by aerobic cellular respiration, it kind of falls within the middle there and has a moderate fatigue resistance. It might about less than 30 minutes. And it doesn't generate large amounts of power, but it does generate a decent or moderate amount of power. So if that's the case then, again, thinking about lifting something, if I want to lift something, which muscle fiber is going to get recruited first? It'll be the type 1. Then after that, if I want to recruit some other muscle fibers that help to generate a little bit more power, not significant, but a decent amount more power, which one would you want to recruit next? This muscle fiber. So this would be the second recruitment. So this one is actually going to have a second recruitment order. So this muscle fiber is recruited second. Okay, so this recruitment order for this one is a second recruitment order. So the motor neurons will go and stimulate the first one. Okay, the motor units will stimulate that first muscle fiber, type 1, first. But as we try to carry a heavier load, what are you going to engage? You're going to engage your type 2a muscle fibers, who can generate a little bit more power. Okay. Now, what kind of activities would this be good for? This is good for very, short bursts of energy, okay? But they can last a decent amount of time, about 30 minutes. Now, I don't know anyone who can sprint along 30 minutes that distance, but it can go, a decent amount of time. So for this case, if someone is actually having this type 2a muscle fibers and you're trying to figure out the main activities that it's involved in walking. So a lot of the muscles that are involved in walking, as well as sprinting. Okay, so as well as sprinting. So walking and sprinting are going to be two big activities that are going to be dependent upon this muscle fiber. So walking and sprinting utilizes these muscle fibers. And again, I'm not saying that every person sprints, 30 minutes. It's just anything less than 30 minutes, but nothing less than a minute. Okay, because if it's less than a minute, it's not going to be utilizing this muscle fiber. It's going to be utilizing this muscle fiber. So it has to be at least greater than a minute, but less than 30 minutes for it to be classified under the activity of this muscle fiber. Okay, that's our type 2a. Now we'll talk about the last one here, which is type 2x. Type 2x muscle fibers, or your fast glycolytic muscle fibers, are very interesting. So let's go back to the structural concepts of it. Okay, well, the first thing that we should understand is, let's look at it first. Okay, that one's the smallest. We already said that one was intermediate, or decent, this one was actually the smallest. This one was the largest diameter muscle fiber. We said that this one is the right in between. So it's intermediate diameter muscle fiber. So we're going to say that this one is intermediate fiber diameter. Okay, and again, I told you in certain animal studies, they found that the type 2x is bigger than the type 2a, but in humans it is not. Okay, so has intermediate fiber diameter. Now, another name for this type of fiber. So again, we said that these are your red, slow oxidative, that is your fast, and technically those are your fast red oxidative muscle fibers. These are your fast white glycolytic. So let's put that word up here. This is the white fibers. And we'll explain why they're white now. Okay, so if you think about it, and we shouldn't even really say white, we should say even palish pink. Let's even add on to this. Let's say palish pink. So palish pink. Okay, just not a lot of reddish hue to it. Okay. Now, why is that? Look at the actual capillary density here. Very little. Look at this. Okay, very little capillary density going into this muscle fiber. So there's not as much capillaries supplying this muscle fiber, then what can we say? Is it going to be delivering a lot of oxygen to this actual tissue cells, this muscle cell? No. So very little oxygen is being dropped off at this tissue as compared to these tissues. Okay. Well, that's interesting. So again, we said intermediate fiber diameter. We said we said very low capillary density. And again, if it has a very low capillary density, what color would you expect it to be? Well, it's not carrying a lot of red blood to it, right? It's not carrying a lot of blood to it. If it's not carrying a lot of blood to it and it's a decent diameter muscle fiber, it's not going to produce very much red or pink at all. So it's going to be very white, or like we said, palish pink, because there's not a lot of blood going to that area. Okay, and then mitochondria, I'm going to put down for right now, very little. Okay, so very little mitochondria, and we're going to understand here why. Well, there's very little oxygen being delivered to this area. So what would be the need for having significant amounts of mitochondria? So if there is very little mitochondria, then this isn't going to be usually utilizing a lot of oxygen. So the amount of aerobic pathways that are occurring here are almost none. So very little aerobic cellular respiration, if any, is actually occurring within this muscle cell. So if there's very little oxygen, if there's very little mitochondria, then aerobic cellular respiration is almost null. Okay, almost no aerobic cellular respiration. So it must be depending primarily upon anaerobic. And anaerobic, we already know. Okay. Same thing with this guy, metabolically. Remember how we said there was a lot of glycogen granules in that guy? There's a lot of glycogen granules in this guy too. So this one's also going to have a significant amount of glycogen granules. And again, what are those glycogen granules called? They're called glycosomes. And what are they going to have in them? They're going to have polymers of glucose, which we refer to as glycogen. And what's it going to do to that glycogen? It's going to break that glycogen down into glucose, right? That's called glycogenolysis. But then what can happen to that glucose? It can be converted into pyruvate. What happens in that step? You produce a little bit of ATP, right? So you can produce a little bit of ATP, but not a significant amount of ATP. But you're going to be carrying out these activities, very fast because it's a fast glycolytic fiber. What was the other mechanism that we could produce ATP anaerobically? Remember this enzyme here? It was utilizing the creatine phosphate, which we denoted as CP, and it was transferring the phosphate from the CP, or the creatine phosphate, to who? It was transferring it to ADP. And whenever these two reacted, what do we get? We got ATP, but it doesn't produce a significant amount of ATP. It only produces a little bit of ATP, right? And again, what was the name of this enzyme? This was the creatine kinase enzyme. Okay, so now, if it's utilizing this creatine kinase enzyme, so this creatine kinase enzyme is involved in this muscle fiber, and also glycolysis is involved in this muscle fiber. So there's going to be glycolysis, and there's also going to be this creatine kinase pathway. So, okay, what two pathways are involved in this one? Okay, well, first off, is it undergoing aerobic cellular respiration? No, it's primarily anaerobic conditions. So it's primarily anaerobic. And by what type of anaerobic process? Creatine, I'm going to put CP pathway, and glycolysis. Okay, glycolysis. And the reason why is because it's doing this is because there's very little mitochondria and very little oxygen. Okay. Oh, real quickly, I didn't even think about this, and I'm sorry. What about the myoglobin content in this guy? What about the little myoglobin dudes? What about those proteins? This one has a decent amount of myoglobin also. Okay? Another thing about myoglobin that we could also say is that myoglobin is also it has a heme pigment in it. And so because it has a nice amount of heme pigment in it, if it's bound to oxygen, what can that also do? It can also produce a reddish hue. So myoglobin also contributes a little bit to the actual reddish hue of the actual muscle fiber, okay? As well as the capillary density. But in this guy, this type IIx, it has very, very little myoglobin. Look, he's crying because it doesn't have a lot of myoglobin dudes to hang out with. So there's very little myoglobin present in this actual muscle fiber. So very little myoglobin present within this muscle fiber. And so that again, that also contributes to the very little oxygen, as well as it also contributes to the very whitish, palish, pinkish color of this actual muscle cell. Okay, now, another thing. This muscle fiber is intermediate in diameter, right? Okay. Also, this pathway, anaerobic pathways, anaerobic through the creatine phosphate pathway and the glycolysis pathway, they occur at a pretty fast rate. So because they occur at a pretty fast rate, they're also going to produce ATP very fast, but just not enough of it. Okay. What about the myosin ATPase activity in this one? The myosin ATPase activity is off the charts. It's insane. So the myosin ATPase activity, I'm not going to draw that whole thing again because we should already know. But this enzyme who's doing what? Breaking down ATP into ADP and inorganic phosphate, which is again connected to the myosin protein. His activity is going to be very, very fast. So if he has a very fast myosin ATPase activity and we're producing ATP very fast, wouldn't this produce a very fast contractility? Yes. Okay, so what do we say here? There's going to be very fast contractility. Actually, the fastest contractility, right? And then there's going to be the highest amount of myosin ATPase activity, right? And then what else did we say? Okay, we said it has fast contractility, high amounts of myosin ATPase activity. It's also producing ATP very fast because it has to only undergo anaerobic, which is a shorter pathway. Another thing, because it's contracting so fast and it's undergoing anaerobic conditions, so it doesn't have a lot of oxygen. It's contracting really fast, so it's going to wear out really fast. What do you think about these actual fatigue resistant with respect to this muscle? Very, very low. So very low fatigue resistance. This is the one that lasts for maybe about less than a minute. So about less than 1 minute. Okay, but this is the one that can produce the largest amount of power. Okay, so because it is anaerobic, doesn't produce a lot of oxygen, but it produces very quick. It's going to contract fast. Also, it's going to contract fast because it has a high amount of myosin ATPase activity. It has a very low fatigue resistance, okay? Very, very low. And the reason why is because it's not producing a lot of oxygen. I mean, it doesn't have a lot of oxygen, so it can't produce lots of ATP. Also, it's contracting really fast, so it's going to wear itself out really fast. Now, it can produce the largest amount of power, though. So whenever you're carrying a load that's extremely, extremely heavy and you're trying to be able to move that load, this would be the last type of muscle fiber that you would recruit. So this has the third recruitment order. So this is recruited last, or in this case, recruited third. So whenever you're looking at muscle fiber types, you recruit, first muscle fiber is going to be type 1. This is the first one that you're going to recruit. The second one that you're going to recruit is type 2a. And the last muscle fiber that you'll recruit is the type 2x, because this can generate the most power. Least amount of power, the moderate amount of power. Now, function for this muscle fiber, again, it's going to be carrying out trying to lift heavy loads or generate lots of power very quickly. So this would be, for example, hitting a baseball, or trying to get your gains on, doing bench press and stuff like that, lifting weights. Okay, so lifting weights is a good one for this one. And also, like I said, hitting a baseball. I'm just giving an example. But it's just very quick in bursts of power, okay? These muscles will be utilized. Now, one last thing I got to talk about, and then we're done in this video. And I'm sorry that it's long. I know I appreciate you guys hanging in there with me. But I want to talk about these muscle fibers, because what can happen is that in one muscle, that bundle right there, this fascicle, right? I might have different types or amounts. So for example, let's say that in my muscle fibers versus whoever is watching this video, I might have a little bit more of the type 2x and the type 2a. Whereas compared to someone out there who runs marathons and is very, very active in endurance activities, they're going to have more type 1. Now, there's two reasons why that person who has a marathon runner will have more type 1s and as compared to me. First off, I don't like to run at all, so I'm not going to have a lot of type 1 muscle fibers. But that's just because these things are contributed by two concepts. One is genetics. So your genetics determines the abundancy or the amount of type 1, type 2, and type 2x. But also environmental stress. What do I mean by that? Okay, let's make this as simple as possible. Let's say that I decide to become a runner for whatever reason, which would never happen. But anyway, if I decided to, I'm going to start running a lot. And as I start running a lot, what's going to happen to these actual muscles? Well, the first thing that's going to happen is, if I'm running a lot, my muscles are going to need a lot more oxygen because I'm carrying out a long activity. If I need a lot more oxygen, what will I need to make more of? I need to make more blood vessels. So the first thing that's going to happen here is I need more blood vessels. So if I'm trying to do endurance, let's say I'm trying to do endurance activities for whatever reason, I'm going to make more blood vessels. So more capillaries. Why would I make more blood vessels? So that I can bring more oxygen to the muscle tissue. Second thing, I want my mitochondria to be big Bertha size. Okay, I want to have a lot of mitochondria, and I want to have big amounts of mitochondria. So I want my mitochondria, because if they're bigger, they're going to be able to carry out more activity. So I want my mitochondria to increase in number and to hypertrophy. Hypertrophy. And the last thing I want, what is the purpose of these actual myoglobins? To hold on to oxygen, to deliver as much oxygen to the tissue as possible. So when blood is coming by, what does the actual blood do? It drops off oxygen to the myoglobin so that whenever the muscle is relaxing, it can drop that oxygen off, produce ATP right? And whenever the ATP is produced, this muscle will be able to contract very heavily, right? Or carry out a long activity. So for this case, I'd want to produce more myoglobin. That's what happens in these individuals. Now, if you think about it for a second, if I make more blood vessels, I have my mitochondria get bigger and I have more myoglobin. Aren't I really, all I'm doing is really switching from a fast oxidative to a slow oxidative? Because that has a lot of capillaries, but this one has a decent amount also. That has a lot of mitochondria, and the mitochondria, you know, decent size, but they're bigger and there's more mitochondria. This one has about a decent amount of myoglobin, this one has more myoglobin. That's all I'm doing is I'm switching. So whenever you go from endurance exercises, you're switching from type 2A to some of the type 1s. That's it, okay? By making more blood vessels, making more mitochondria, having hypertrophy, and having more myoglobin. Now let's take the contrast. Let's take the opposite. So let's do that down here now. Let's say that we're doing the good stuff. You're trying to join the gain train, all right? You're trying to go from a puny human to a, fragging Hulk. So if that's the case, then I want to start doing resistance exercise. If I'm going to start doing resistance exercises, I'm going to start trying to lift heavy loads. As I try to lift heavier loads than normal, I put a lot of stress on my muscles. And three things happen. When you put a lot of stress onto the muscle, your muscle fibers actually split. Let's say here I have a muscle fiber. I'm looking at it like this, right? Let's say here's this muscle fiber. And this muscle fiber, what can happen is I can split this muscle fiber longitudinally from excessive amounts of weight load. When it splits, something really crazy happens. You have these cells. They're called myosatellite cells. Myosatellite cells. What these myosatellite cells do is they come in and they plug and proliferate and fuse into that little longitudinal split and grow into it. As they grow into it, guess what happens to the size of this actual muscle fiber? It gets bigger. And as it gets bigger, it starts, what is that called when a muscle fiber gets bigger? It's called hypertrophy. So that's one thing that's going to happen, is whenever the muscle cells have a longitudinal split, the myosatellite cells proliferate and plug into that and fuse in there and develop a larger muscle size. That's one theory. The second theory is, what is making up a muscle fiber? What did I tell you was one of the main components of this muscle fiber? If I were to pull one of these muscle fibers out, I told you that there's thousands upon thousands of these things called myofibrils. I want you guys to just think for a second. If I make more myofibrils, isn't that gonna, myofibrils are actually occupying 80% of the cell volume. That's a lot. So if these guys, if you make more of these or you make them bigger, what's gonna happen to the actual cell volume? It's gonna get bigger. If the cell gets bigger, what's gonna happen to the overall size? It's gonna hypertrophy. Okay, that's one thing. As well as, think about mitochondria. If my mitochondria get bigger, or I have more mitochondria, that'll also make the cell bigger. Also, another thing is those glycosomes. If I have more glycosomes, I'm gonna have more glycogen. If I have more glycogen, the muscle cell is gonna even get bigger. And the last thing that they believe can happen with the resistance exercise, besides the myosatellite cells fusing this longitudinal split and making the muscle bigger, as well as increasing the number of myofibers, as well as increasing the number of glycogen, as well as increasing the number of mitochondria, they also believe that you can switch some of your fibers. Particularly, you can switch some of the type 2A fibers into the type 2X fibers. And those are for more of the actual heavier resistance exercises. That should make sense. All right, so the last thing I want to mention, guys, is disuse atrophy. It's really simple. Think about it. Whenever you're actually not utilizing a muscle, what happens? It starts actually decreasing in size, because if you don't use it, you lose it. That's the whole concept. So what can happen is, if you do resistance exercise, think about a bodybuilder who's been lifting for a while, spent years and years working hard to get those big, nice muscles, right? And he switched a lot of the type 2A to a lot of the type 2X. Let's say that for some reason, he's like, I'm done bodybuilding. I don't want to do this anymore. What will start happening to that person's muscles? They'll start decreasing in size. When they start decreasing in size, what is that called? That's called atrophy. And sometimes that can happen due to an injury, or sometimes it could just due to not wanting to do anything. But that's one of the important things, is that if you do not utilize a muscle enough, the actual muscle fiber can switch back from type 2X to type 2A. And then again, whenever the muscle atrophies, it becomes weaker. And if the muscle becomes weaker, that can actually be a bad thing, okay? So overall, guys, I really want to say thank you guys for sticking in there with me. If you guys watched this whole video, I appreciate it. I hope it all made sense. That's our goal here at Ninja Nerd Science, is to make it make sense, and I hope it did. If you guys did like this video, please hit the like button, subscribe, put some comments down in the comment section. All right, Ninja Nerds, until next time.